anaplastic glioma, a rare type of brain cancer. Lead author, Dr. Martin van den Bent, is a professor of neuro-oncology at Erasmus MC Cancer Center in Rotterdam, the Netherlands, and he will be presenting this study. Professor van den Bent.
Ladies and gentlemen, it's my pleasure to report on behalf of the EORTC Brain Tumor Group the results of this trial that will be practice-changing. It's the CATNON trial, and the trial set out to investigate the activity of temozolomide in 1p19q non-codeleted anaplastic glioma. What are anaplastic astrocytoma? Anaplastic astrocytoma are a subgroup of the gliomas. It's an orphan disease. There are approximately 1,500 new cases diagnosed each year in the United States. We set out for this trial because in previous studies we had made two observations. First of all, the EORTC identified temozolomide as a major component for better treatment of glioblastoma patients. We conducted a trial in the early 2000s that showed that if you combine radiation therapy with temozolomide and you follow that with another 6 cycles of adjuvant temozolomide, survival is increased. Glioblastoma is a chemotherapy-resistant disease. At the same time, we did a very similar study on adjuvant chemotherapy in another subset of anaplastic glioma, the so-called oligodendroglioma. We made in that trial the observation that adjuvant chemotherapy did not improve outcome, whereas that disease is to be considered chemotherapy sensitive. So that was strange. We also noted in that trial that there was a particular subgroup of patients with a particular worse outcome, the patients without the 1p19q codeletion. We already knew at that time that 1p19q codeleted patients were much more sensitive to chemotherapy than the patients without 1p19q codeletion. So we asked ourselves whether if we would give temozolomide to these patients without the 1p19q codeletion, whether that would improve their outcome. And we set out a little bit complicated trial, especially if you consider that this is an orphan disease. So we asked ourselves basically two questions. Will temozolomide given during radiation therapy improve outcome in these patients? And the other questions we asked was whether temozolomide given after radiation therapy would improve outcome. Primary endpoint of this study was overall survival. We required for inclusion in the study that the central laboratory confirmed the presence of an anaplastic glioma and of the absence of 1p19q loss. So it was also, from the logistical aspect, a complicated study. Samples had to be sent before patient inclusion. And we randomized our patients into 4 arms: radiation therapy alone, radiation therapy plus concurrent temozolomide during radiation therapy followed by 12 months of adjuvant temozolomide, and then radiation therapy with both concurrent temozolomide and adjuvant temozolomide. On Sunday morning at 8 o'clock at the CNS symposium, I will show the outcome of question number 2. So we are comparing basically arm 1 and 2, in which no adjuvant chemotherapy was given, to arm 3 and 4, in which adjuvant chemotherapy was given. Why are we doing this? Well, it was a complicated trial, like I said, and this is an orphan disease. So it took a collaboration of many cooperative groups to enroll the number of patients that were required for the study. We needed 750 patients. To get those 750 patients, we screened no less than 1,400 patients in 118 institutions, 12 countries, and 3 continents. We had a planned interim analysis after 41% of events would have occurred, and that was in the summer of 2015, just after the completion of enrollment. And we did not anticipate anything to come out of this interim analysis, as usually these trials take a long time to produce positive results. But we were completely taken by surprise when the IDMC that looked into the data recommended the release of the data of adjuvant temozolomide. We had at no point in time anticipated this outcome. Why did they recommend this? Well, here you see in univariate analysis the outcome of the study for adjuvant temozolomide treatment only. In red, you see the patients that were not treated with adjuvant temozolomide. In blue, you see the patients that were treated with adjuvant temozolomide. What comes it down to in figures? Well, if you look at the 5-year survival, that increases from 44% without adjuvant temozolomide to 56% with adjuvant temozolomide. That corresponds to a hazard ratio of 0.67 and a highly statistically significant p-value. So what do we know now? We know now that temozolomide given after radiation therapy improves survival in this disease. By this trial has become a practice-changing trial. We need further follow-up for the question number 2, as the IDMC made no recommendation about that part of the treatment. So some of the results of this trial are still unknown to us, and it will take more years of follow-up before we can make definitive statements about that the contribution of that part of the treatment. What is also for us quite important to emphasize is that this is a rare disease, and it requires trials of many, many years duration. So it's a privilege to have been working with a number of groups that made this possible. We had EORTC and its partners in the UK, in Australia, in the United States of America and Canada to work with us to make this possible. But you can imagine that it took quite an effort to make the results of this trial available in an orphan disease like this. Thank you for your attention.
Thank you, Dr. Van den Bent. I mean, I think this illustrates very helpfully the theme of this year's meeting, which is collective wisdom. And even more than that, it illustrates that even within a rare tumor type and using standard chemotherapy, it is very important, and we can select patients who are most likely to benefit from treatment, and at the same time, potentially spare patients from the toxicities of therapy that they are unlikely to benefit from. So congratulations for this.